It is my pleasure to introduce our third speaker for this session, and that is Doris Wang from the University of California in San Francisco. Dr. Wang is an associate professor of neurological surgery at UCSF. She's also a Chen scholar, so we're very excited that she represents the Chen Institute here today. Her clinical focus is using neuromodulation and ablative therapies to treat movement disorders. And her research interest is to understand the brain network dynamics that control complex motor behaviors, such as gait, and using novel neurostimulation therapies and strategies to improve motor functions in disease states. So we will hear more about walking through the everyday world in her talk, and I'm very much looking forward to it.
Thank you so much, Matthias. And I would like to thank the organizers for this symposium. Such an honor to be here. And I'd like to also thank the Chen Institute for supporting me as an inaugural class of Chen scholars at UCSF, and also for co-sponsoring the symposium. So the talk today will be walking through the everyday world, how understanding the neural substrates of walking, and how we can use neuromodulation to improve walking for patients with Parkinson's disease. These are my disclosures, none of which are really relevant for the talk today. So walking is a fundamental motor task that most of us take for granted. It mobilizes us from point A to point B, allows us to stop and start at will, change directions or gait patterns to avoid obstacles, and also allow us to maintain balance so we don't fall. These are just some little videos of my younger daughter walking. However, for patients with Parkinson's disease, these simple tasks that we do thousands of times every day becomes a huge burden because gait and balance impairments are a major, don't know what happened there, are a major problem for these patients. They may manifest as difficulty in initiating walking, difficulty in maintaining reciprocal rhythmic locomotion between the two legs, or adjusting step lengths or body positions to perturbations in the environment. And unlike other Parkinson's symptoms, which you see as tremor, bradykinesia, rigidity, advanced gait disorders, like the ones I talked about, or freezing of gait, where the body suddenly stops moving, are notoriously unresponsive to traditional therapy like medication or deep brain stimulation. So the goal of my research is, one, to understand the pathophysiology that underlie these gait mechanisms or dysfunctions on a circuit level, and also, two, using advanced neuromodulation therapies to kind of close the loop and fill this critical need in clinical treatment. And one of the main challenges in treating gait disorders for PD patients is that the neurocontrol of gait in humans is actually not very well understood. And from a biomechanical perspective, human gait is an extremely complex motor task that requires a precise coordination of movement across all four body limbs, besides the trunk, while we're trying to maintain balance. And we're one of the only species that's bipedal. So, the studies we do in cats, quadruped animals don't really apply. And one of the other things is that, during continuous locomotion, right, basically the body has to generate these alternating movements where one leg is in contact with the ground while the other one is in the air. So that's called the swing phase and stance phase, which I will go into detail a little bit later. And, when I was starting to look into this field, a lot of this literature is derived from animal models, non-invasive studies, kind of in the fMRI, which, we're not really actually studying gait, but imagine gait, or from externalized leads or, EEG systems, which, is burdensome when we're not actually navigating the real world. And based on this collective line of study, the hierarchical control, supraspinal control of gait, is thought to start in the area of, I don't know if the laser pointer works, but start in the cortical areas of the SMA, supplemental motor area, premotor area, and M1, where these they descend down to the basal ganglia for refinement. And then they kind of send projections down to the brainstem centers, the locomotion centers where cerebellar inputs join in before everything goes down into the spinal cord for output. And disruptions in any part of this process can cause gait dysfunction. And when I was in my training, one of the ways we try to study gait in humans are in the operating room. So when I do deep brain stimulation surgery, we used to do these awake. And that's a great opportunity for us not only to record local field potentials, which are pooled subthreshold neural activity from hundreds or thousands of neurons, but also gives us the opportunity to put in an electrocortical strip covering different cortical areas. So from there, we can now study, kind of the cortical, subcortical control of stepping behavior. Again, this is not real gait, but gives us an insight into, what the brain is doing in terms of coordinating these intralimb behavior. And as I was coming out of fellowship, there's a revolution in types of technology that enable us to actually do study this in people who are freely mobile and moving. So now we have this generation of bidirectional neural implants, totally implanted, that not only sends stimulation to basal ganglia or other parts of the brain, but allows us to record and stream data wirelessly. So it gives a really awesome platform to study these natural behavior without constraints of electrodes and tethers and wires. So using this, hopefully the video can play. All right, so using this technology, I started to study how Parkinson's patients walk. This is kind of in our lab setting, so where we outfit them with wearable sensors. So use a combination of IMUs, inertial measurement units, as well as force-sensitive resistors, goniometers to capture gait kinematics. And in the research device, I will go into a little bit further, we actually have the ability to synchronize the accelerometer, which is built on top, within the device itself, with our kinematic measurements. So from there, we can then do offline analysis of neural control of gait and balance. All right, so today I will focus on two main projects in the lab, which is using adaptive closed-loop neuromodulation to improve gait. So one of the things, we do, or I do as a neurosurgeon, is, implant DBS for patients with Parkinson's disease. And it works really well at controlling some of the other symptoms. And DBS, if you don't know, works by continuous stimulation. So basically, we typically use really high frequency, 130 to 180 hertz, with a predetermined frequency pulse width amplitude is set by the patient's neurologist. And the theory that it works is by it desynchronizes pathological network oscillations. So work by many groups around the world have shown that different symptoms and signs of Parkinson's disease is related to highly synchronized or pathologically synchronized frequencies in different ranges, like theta for dystonia, alpha for tremor, beta for bradykinesia, and even narrowband gamma for dyskinesia. That's too much unwanted movement. However, as everybody knows, gait is a very dynamic movement state. So the brain is probably not in a static state, like stuck in a very pathological frequency band. While DBS works really well at abolishing beta for Parkinson's, the cost of that is actually probably not letting the physiological, transient, and natural synchronization that happens during gait to occur. So one of the first ideas I had was whether we can use an adaptive algorithm where we can record activities from people's brain as they go about doing their normal overground walking, classify left versus right steps or leg swing, and using stimulation to ramp up or down stimulation in accordance with their gait cycle. And that may restore kind of their natural patterns of gait. So to perform this, I was actually fortunate enough to recruit 5 patients with Parkinson's disease who are candidates for DBS surgery, but also they had off-medication gait problems. So they can manifest asymmetry, freezing of gait or slow gait, but with medication, their gait improves some to a degree. And so these patients were implanted with mostly bilateral. One patient only had a unilateral implant. Palatal DBS lead targeting GPE, GPI. Simultaneously, I implanted permanent ECOG strips overlying the premotor and motor cortex of these patients. And for electrodes from each hemisphere is connected to a research version of the bidirectional interface called Summit RC+S. This is not the commercially available one, which actually offered us a lot of advantages. In fact, we had full access to the application programming interface. So with these patients, we recorded a ton of data, hundreds of hours of data, both in the lab and at home. So the first thing we try to classify is, can we actually detect neural signatures of left versus right leg swing from these patients? And majority of this work is done by my former postdoc, Ken Louis, so I will be highlighting his work here. So this is just one example of a single patient. These are kind of rastergram, single trial. Each line represents one single gait cycle from left heel strike to left heel strike. And it's very small, but there are little color dots indicating right heel strike in there as well. And the left is showing palatal alpha frequency activity. Warmer color means higher amplitude, and then the other one is N1 beta. You can see in the low medication state, so when a patient's not taking his Parkinson's meds, gait step length and time is very variable, which is a marker of Parkinson's. However, when the subject takes medication, you can see by the rastergram, basically the variability decreases. Each gait cycle is much more uniform. It's much more similar. And what's also concurrent with that is you can see that the colors of the blob, so basically the amplitude of alpha signal in the pallidum and beta seem to be more synchronized during specific phases of the gait cycle. So using this data, we actually did a more comprehensive grid search in which we look at each single power band and combination of power band to identify the best power band within each hemisphere, whether that's coming from the pallidum or the cortex that best predicts the gait cycle. And this took years of work, but kind of summarizing here, can we actually feed these neural features on board the RC+S LDA classifier? And changing stimulation amplitude on a really fast time scale. This is within a few hundred milliseconds as the subject is walking. So I'm going to start to play this animation, and this is slowed down 3x because each gait cycle is about 1 second. So on the upper panel, you see the left hemisphere stimulator, and on the right is on the bottom. The teal and purple colors are actual phases of the gait cycle for right versus left leg swing. And then the black line is a state classifier based on the Summit device, whether they think it's, one is actual swing state based on a predetermined threshold we set, which is in the dotted line. And then the red line is actually the constant ramping up and down of stimulation amplitude for that particular patient. So while it's not tracking perfectly, but you can see that the two hemispheres are off cycle, which is what happens during our natural gait cycle. So we're able to independently control stimulation amount to each hemisphere based on the patient's natural gait cycle. So this is actually a huge technical achievement. And now the question is, now that we can implement it, does it actually have an effect on their gait? So we perform a bunch of blinded testing. So the subject themselves don't know which setting we're doing, so whether continuous clinical DBS or with their adaptive DBS. And this is based on a palatal biomarker. And these videos are played at the same speed. And you can see, side by side within the same day, adaptive DBS settings seem to make the patient walk a little bit faster. But again, we did it systematically across 5 patients, all 5 patients. And I know it's a really busy slide, so we kind of present each patient's step length, step time. Left foot is showing in kind of the salmon color, and then right foot is in blue. Again, there's a lot of variability. Some people, have longer step length. Some people have, faster step time. But what's actually pretty I guess, common across all subjects is that in general, variability in terms of step time or step length improved with adaptive DBS, which shows on the right column compared to the continuous DBS. What's more interesting is the symmetry. So what this pattern stimulation does, and if you just can focus on the panel G, which is the pooled step length or step time symmetry across all subjects. Adaptive DBS, it seems to restore the symmetry between the left and right leg during walking. So this is done during the clinical setting, after many months of optimization. What about in the home? So once we establish the safety and tolerability of these settings, we then embedded, we performed a double-blinded, multi-day evaluation comparing 2 different adaptive settings for each patient compared with a clinical setting. And basically, subjects on these settings were about 7 to 10 days each, and they're completely unaware of which setting. So we have a ramp up in which the baseline is half their stimulation amplitude. So as they're about to take a swing with their contralateral leg, the stimulation amplitude ramps up. And ramp down is basically going from their constant setting, kind of dropping down stimulation. And based on this, again, these are after a week or so of chronic adaptive DBS at home. We brought them back into the clinic, again, looking at step length, step time. And in certain, across patients, we only had 3 patients who enrolled in this long-term study. Again, step length variability seemed to improve, and most of the patients actually improve more with this ramp down setting. So again, suggesting that perhaps constant high stimulation, high frequency stimulation may be detrimental to gait. So if we're actually introducing more entropy by ramping down amplitude, we can kind of restore some of these natural phenomena of gait. And in terms of the patient's subjective ratings, so again, each subject is across rows. So we ask them, so compared to your normal clinical setting, is adaptive setting better, worse, or same as your clinical setting? And on the left side is all their regular Parkinson symptoms. On the right is actually more gait-related metrics like falls and freezing of gait. So in general, adaptive setting wasn't too detrimental. They usually rated about the same as their clinical setting, except one patient. The last one, she's really mild and she's tremor dominant. So she did detect a slight difference. And that on some of the days, the adaptive setting seems to be worse in her clinical setting. But in terms of the top 2 patients who actually has a lot of freezing of gait and falls, in some of the adaptive settings, it seemed to actually reduce the number of falls for subject 1 especially. And then subject 2, it seems to be also subjectively better. And actually, both of those patients voluntarily choose to remain on their adaptive setting even after the double-blinded period. So this gives a lot of promise for using, this fast-paced, gait-phase synchronized adaptive DBS to perhaps help patients' gait disorders. Not everybody. So the reason the other 2 patients didn't enroll in this, multi-day long-term study is because we couldn't identify biomarkers that switched a lot, And so for this to work, we need reliable biomarkers. So we thought of another strategy, for what we call a slow adaptive DBS algorithm. For this, basically, we want to systematically identify stimulation parameters that improve gait. And this may be a lower frequency, lower amplitude, different, again, we're working within the constraints of the system. So we want to use a data-driven way to find that stimulation that improves gait. But again, it might be at the expense of other, Parkinson symptoms. So our idea is to switch that setting only when the patient's walking. So this requires classification of walking versus non-walking periods. So this is kind of a schematic. So the idea is, while the patient's just talking, sitting around, we use their clinically optimized DBS by their neurologist to help treat their tremor, bradykinesia. But when they're standing up and walking, we switch to this different algorithm that's movement state specific. So to employ this, I'm going to highlight work from Hamid, who's actually sitting in the audience. He has a poster about this. And Rithvik, a medical student working in the lab. So the first part of this is how can we use these bidirectional devices to actually identify personalized gait optimized settings within each patient. So if you don't know, after I implant the electrodes, patients basically sit in a neurologist's office and they go through stimulation settings. It's like an hour, 2-hour long process to see which one makes it better. But very few people actually test people's gait, or if they do, it's just 30 seconds a minute. So what Hamid developed is actually a walking performance index. So taking parameters that really has been shown clinically to be abnormal in patients with Parkinson's disease compared with other healthy subjects, which is stride velocity, step time variability, step length variability, arm spring amplitude, and basically create a composite score so that can give us an objective measurement and rating of each patient's gait. And he then systematically changed either frequency, amplitude, or pulse width. And after each setting change, again, subjects are blinded. He did multiple measures within each clinical setting. Then using a Bayesian process regressor, he basically predicted, what setting that may predict improved walking performance versus settings that worsened it. And you can see that, based on this example, even after, visit 2, which is, after testing about 8 different settings, we're able to come up with a stimulation parameter that is optimized for that individual's gait. And then again, he brings back the patient and validates it. Again, this is a paper just came out in NPJ Parkinson's disease if you want to look at the details. And what's more interesting besides, the kinematic measurement is actually we have access to these patients' neural data as well. So then Hamid looks at, what is a common theme? So some patients walk better at 60, 70, low frequency stimulation. Some people walk really well at 190 hertz. But what's common amongst all 3 subjects after repeated measure is that those parameters that seem to improve walking actually had a common neurological or neurophysiological signature. So during specific phases of the gait cycle, meaning and in this case, during the double support period prior to contralateral leg swing, you actually saw decreased beta activity. And also this carried on during ipsilateral swing period as well. So not only did we characterize kind of a systematic data-driven way to predict settings that can improve individual gait performance, now we have a physiological basis. So now using collective data and AI, we can maybe derive at this parameter a lot faster, even when the patient's at rest. That's kind of the goal. So now that we have the gait optimized setting, how do we implement it? So for this, Rithvik, who's a medical student working with me during his summer project, basically aligned manually hundreds of hours of data from patients when they're walking at home. So for all the study participants, before we turn on their DBS, after they're optimized too, during different time periods, we ask them to wear these kind of ankle accelerometer devices called Rover, which tracks, when they're walking, the directionality, and, pretty well-validated markers of walking versus non-walking. And basically, Rithvik aligned the Rover accelerometer with the onboard accelerometer of the neural device, and basically try to figure out the neural signatures of walking for each individual subject. And down below is that when we bring the patient back, there's really high concordance and validation of the Rover. So when the Rover says, this is the period when the subject is walking, it actually correlates super well, like 98% sensitivity and specificity. And this is an example of, just kind of grand average power spectral density graphs, signatures of periods when patients are walking, which is shown in blue, versus non-walking periods. And you can see that the spectral power is pretty different. And this is over multiple hours across different medication cycles. So this is really powerful data in knowing exactly when the subject is walking versus not walking. And these are what he's showing, the individuality, right? There's no one size fits all based on patients' physiology, symptom presentation, and their brain neurophysiology. We know that precision medicine is really important, and this just shows that even though I tried to put the electrode in the same spot in each patient has a distinct feature importance of spectral bands that best predicts walking versus non-walking. For some subjects, like subject 1 on the left here, the palatal shows a really great biomarker and has a higher feature importance rather than the cortical, whereas for subject 4, M1 shows the best predictive power in terms of determining walking versus non-walking. And even within the subject, the left and right hemisphere shows quite a bit of difference as well. So Ritwik, again, using the LDA classification, and we're using LDA predominantly because the device, the Summit RC Plus S, uses that to determine their state classification for the onboard closed loop algorithm. So again, we're able to achieve with pretty high fidelity about 80% AUC. To 90% in terms of determining walking versus non-walking. And this is using different biomarkers from different brain regions. So this is just an offboard. So basically what Ritwik did is using simulation, kind of fed back a period of neural data into the simulation to determine walking versus non-walking. And the middle panel there shows the adaptive DBS state based on the biomarker and the threshold he sets. What's super exciting is just, kind of a month ago, Ritwik and Hamid actually tested this out in real time in patients in the clinic. So, we have the patients do a variety of things, sitting around just waving his or her arm, moving things, walking, sitting up in chair and standing, a variety of tasks, and navigating around the lab space. And the green are the true positives, and then the bottom, basically 0, would be the gait optimized setting. And then the 1 is the other setting. And you can see it actually has pretty good sensitivity in picking up all the periods of walking. There's some false negatives, or false positives, I should say, but overall, generally, it's working really well. And subjects themselves actually felt great on these settings. And this is just an example of their walking performance. So green is our gait optimized setting, what it looks like. Gray is their clinical setting, and then blue is in the adaptive setting. So looking just within the periods, they seem to do quite well. So we're really excited about this. We're about to start our first patient doing this at home, I think next week. So, based on this data, hopefully I've convinced you that adaptive DBS is a really powerful tool to not only understand the neurophysiology and control of gait, but we can actually directly implement the knowledge we gain to hopefully improve gait functions in Parkinson's patients. We can run it on a sub-second accuracy on a really fast time scale, or we can do it on a slower time scale based on home biomarkers. And then in the last few minutes, I'd like to show a more interesting study about swimming. So I've shown you about walking. Of course, it's really important, but we have one subject who is an avid swimmer. He still swims despite his Parkinson's. And this work is done by Shravani, who is a MD, PhD, also neurosurgery resident in our department. So basically, Shravani rigged up this system where we're transmitting wirelessly via Bluetooth, streaming neural LFP from this subject while he's swimming in a swimming pool. We asked him to do, backstroke is better for signal transmission, so that's why we're looking only at backstroke. So he did just kicks, just arm, strokes, and then Shravani actually placed, these waterproof accelerometers on the wrists and the feet of the subjects. And we have the subject perform similar stroke movements in air, which is shown kind of on the top, and similar kicks he would do on land versus stepping and walking naturally in panel C versus, what he does in the water. So kind of the question is, taking gravity out of the equation, less impact, how does the neurosignature change? And also swimming, staying afloat, requires probably higher level of coordination. And then thirdly, just why not? Now we have these tools, and this is what these devices are kind of built for, right? To learn about neuroscience out in the natural world. So I don't have a lot of time to go into details, but what you can see here on the left is, basically time locked to periods of maximal acceleration on the arm swing. for swimming and then walking on the right. And you can see the activation patterns, and this is looking at the cortical activity, are quite different. So for swimming, you can see a lot more gamma activity, which is correlates of spiking, whereas walking, there's more lower frequency modulation. And then when you look at within each trial, kind of the modulation index, how much the amplitude varies, time sync to the basically the period of maximum movement. Between swimming and walking, this is again looking at cortex, blue is swimming, black is walking. So there are different modulations, even though the electrodes are in the same location. Even for arm swing movements are similar on water and inland, the activity patterns are quite different. I won't go into the details. And then for the pallidum, you see the same thing. And then if you look at the pallidal leads, there are a lot of artifacts. That's because of the stimulation artifacts. But if you look at the low frequency activity, there's still quite a difference in terms of low frequency modulation of swimming versus walking. So hopefully I've shown you the power of these totally implantable systems to understand the neuroscience and underpinnings of complex movements in the real world. From this data, we can generate a lot of hypotheses and neuromechanisms to test using targeted stimulation about the planning and execution of complex actions. And again, I think the real power is that, from what we learned, we can then directly implement it and program it into the device to hopefully treat patients and improve their outcome. So with that, I would like to thank my wonderful, amazing lab. Two of the members are here, so Hamid and then Sang, who you'll hear later from about a different project. The collaborators, all my funding sources, and yeah, thank you for listening.
All right, thank you so much. That was very cool. I love the swimming part. I think I actually put in my job application, like, oh, now we can record all these signals when people are like rock climbing and swimming. And
Yeah.
It's fun to see that actually somebody's doing it. It's exciting.
Yeah.
Do we have some immediate questions? Great.
Hi, James Cotton, Northwestern. Great talk.
Thank you.
I think your data on the reduced falls kind of like mitigates this question, but there's good variability and bad variability, right? And there's often like the U-shaped curve with that.
Sure.
And I could easily envision when you're pacing the brain that you actually are kind of suppressing variability, but some of that might suppress useful variability, like as control strategies. So do you have any concerns or thoughts around that?
Yeah, that's a great question. So, what I showed you is actually just straight path walking. We didn't actually put into turns because turning is like another can of worms, right? But in some of the other subjects, again, not part of this study, I have a different system where I can just tell the programmer to pace stimulation with like 100% accuracy. And in those patients, for patients with a freezing of gait, we actually showed that we were able to reduce freezing. So it seems that, at least in limited sample size, again, this is mostly in the lab, it seems to improve gait and falls. Yeah,
Thank you.
Thanks.
I might have missed this, but I was wondering what kind of activity you are sending that's synchronized with the gait. Was that in a specific frequency band, and like how customized was it to each of the subjects?
Yeah, so it's very customized. It spans for some subjects it's just a couple frequency bands, like say 13 to 15 hertz. For others, it's a much wider band, like spans like 30 hertz frequency. And it again depends on the subject. Sometimes it's in pallidum, sometimes the biomarkers in the premotor or M1 area. So very different across individuals.
And then I was wondering if you think that the cortical strips are going to become a more common thing to insert, because it seems like that's a great source of information.
Yeah, absolutely. I think so too. It's not prone to the artifact, as you can see, right? With the swimming, like you have these simulation artifacts and yeah, the cortical strip is more sensitive, but the problem is it's also more invasive. This an added electrode introduces a lot of risks for the patients. So yes, ideally we would have other ways of recording cortical activity semi-invasively, but it's probably not for everybody.
I actually have a quick follow-up question, very similar, I think. So I love the idea of stimulating at particular gait cycles that you mentioned. I know from different fields, like for example, memory enhancement or cognitive enhancement, it's like not just behavioral information to use to kind of precisely time stimulation, but also like specific phase, for example, of the underlying neural oscillation. Does that play a role at all in this? Like, does it matter when exactly you stimulate based on the neural activity?
Yeah, that'd be great to do like a phase controlled, because I think phase amplitude coupling changes dynamically as well. So if we can get to that level of precision, it would be amazing. But I think we're running into kind of the hardware limitations at that point. You need a more distributed system, which is cumbersome and then introduces delay. So yeah, hopefully, to the device people out there, if you guys can engineer things that can capture, coherence, phase amplitude coupling, that would allow us to perform these kind of phase, on top of slow phase, right? Or like burst stimulation. I think different stimulation patterns, there's so much underexplored parameters in terms of neurostimulation that we haven't explored. And that would be extremely useful.
Perfect, thank you. Then I think the next step would be that I'd like to ask all speakers of the symposium to come back on stage, and we have our general Q&A. And at the same time, I encourage everybody to formulate your questions and come to the microphone here and ask questions to all the speakers that might be relevant, maybe for one or two or to everyone. That would be great.
Okay, hello, this is Hasan Ayaz from Drexel University, and I have a question for Dr. Maren Bixen. Well, thank you for sharing your impressive solution for the scalability challenge of neurotechnologies, and you addressed both cost, miniaturization, and user interface simplification. So do you think one of these dimensions is more important than the other? And if you further go deep down in one, you can improve the scalability potential. Like, for example, if you further miniaturize the system, would it help? It looks like you still have potential on all these. And the follow-up on that is, can you comment on the regulatory pathway that you picked for this? Because that is the efficacy demonstration of the overall system. Thank you.
It's a big question. I'll give you a quick answer. We're not worried about miniaturization right now. I mean, the active elements are in the milligram range. And so actually the size is almost all the enclosure. So that's one of all the problems we've had. That's not one of the problems you've had. I mean, the other issue, you really talk about things like cost and scalability. And I think this is really the difference between, let's say, academic-centered research and really thinking about translation. If you're at that laboratory scale, it's okay to make 100 a month, or maybe okay to make 1,000 a month. In this case, scalability means, making 1 million a month. You know what I mean? And so it's a completely different scale of process. And same thing with cost. Again, if you're satisfied staying at that academic stage, frankly, you can make each one for $100, right? As long as the grants allow it. While when you're thinking about translation, obviously, the pressure becomes very, very high to get to sort of a Band-Aid or a pill level. So there's no simple answer to that other than to say that, really with the translation, the pressures are high. And as far as regulatory, as long as we can keep this in the non-significant risk range, we can make that process much simpler. And I didn't talk about it, but a lot of the engineering went into risk management. These things are, I say borderline edible, and then someone says eat it. But they certainly won't, the electrochemistry prevents an excessive discharge.
While others think about their questions, I have another question actually to I think it applies pretty much to all of you. Like, when I think about bringing this, from the lab into the real world, I think what's interesting to me is also like the long-term effects, like of, stimulation or BCI usage. And I find it hard to distinguish, like, for example, things like gait in the course of disease probably like get worse anyways, right? So how can you distinguish like things if the kind of the treatment effect decreases over time? Do you know? So the question is like, do you know if that happens? Does the effect decrease over time? And how do you know if that's because of the disease getting worse, or is that something that is more like maybe technology, like, I don't know, impedances getting worse, or the system kind of habituates or adapts or something like that?
I can take a stab. So for the gait-related research, these patients have been implanted for over 2-3 years now. And actually, Ken did analysis in looking at stability of the biomarkers over that time span. And for most of the subjects, it has been surprisingly stable over a year or so in terms of the biomarker from when he first tried to whittle it down versus the latest visit. So I think, I guess maybe that's advantage of less invasive in terms of like non-intracortical, right? With ECOG and local field potential, the signal seems to be pretty stable. But you're right, in terms of disease course, with disease progression, like their dynamics are gonna change. Yeah, we haven't found a great way of addressing it besides, periodically checking in to see how well these, classifiers are performing.
Yeah, for brain-computer interfaces, for intracortical electrodes, until the last year or two, all of the human work had used that device that I showed you, the Utah array. And so it's a rigid device. It's mechanically mismatched from the brain, and the materials tend to degrade over time. So I think that's every device that's implanted, the signal quality does degrade, and then therefore performance is lost. There are now other devices that are actually in clinical trials led by companies that are using flexible materials or targeting different structures, like even trying to use intravascular approaches. All of these have trade-offs. I think it depends, like what level of information do you actually need to get from the brain? If you just need an on-off signal, you probably don't need an intracortical electrode. If you need that level of movement, though, there needs to be advances in the materials and design in order to preserve that longevity. And just, as the other speakers were saying, as for people who have neurodegenerative conditions, certainly that's you're fighting an additional factor where the actual information that you're able to extract does degrade over time as well.
That was a long one. Hi. Mine's quite a high-level question, but when we think about scalability and neurotechnology, how do we think about how we engage people with this technology? So we kind of tell people, we're going to do this kind of work with your brain, you're implanting deep brain stimulation, for example, and we're thinking about trying to do this over millions of people. How do we engage? For example, I work with a lab in Sheffield who do DBS, and they've had neurologists that have never seen someone from a BME group, minoritized ethnic group. How do we engage people like that in this technology? How can we work with different groups to make them understand what we're doing and how we can integrate it wider into society, and how we're doing that moving forward?
Yeah, I mean, I do think it's a big challenge, right? I was really struck in your talk of there've been therapies that have existed that people just don't adopt, right? Because the actual process of getting them is too challenging. And so that's certainly where we are with brain-computer interfaces. Only a handful of surgeons in the world know how to implant these devices right now. It takes a team of people to calibrate them and to keep them operational. Even in our case, when we do have a home system that people can use, some participants have caregivers who are really willing to learn how to attach the device and kind of get things set up from day to day, and others don't. And so I know that a lot of groups and companies are working to develop systems that are kind of always on, that the person themselves can use, that requires wireless technology that does exist now. But I think that there's still going to be some hesitancy, right? The benefit needs to outweigh kind of the risks and challenges associated with doing that in order for people to start to adopt it. I would say that implanted technology does have lots of applications, just deep brain stimulation, even pacemakers for a heart. Sometimes you do need to implant in the body to deliver the amount of function that you are promising, right? A wearable would be great for a BCI, but we don't have access to the level of movement-related information that we need to restore upper limb function, to restore speech, and so you do need an implanted technology, but it needs to be something that can be scaled. The surgical approaches need to change, the cost needs to be lower. And it needs to work all of the time. Like reliability and accuracy requirements for assistive technology is very high, and so we have a pretty high bar that we need to go after.
Yeah, and just from like the movement disorder perspective, DBS is one of the most underpenetrated and underutilized technology. There's so much class 1 evidence that it works great, yet only probably 10 to 20% of the patients who qualify actually end up getting it. But I think this is a power, in terms of scalability, like a power of industry collaboration, right? Medtronic now, their state-of-the-art generator that everyone's putting in now has this sensing function, and you can activate it. And adaptive DBS, a few months ago was FDA approved. So this is possible. I think as it generates more interest, and there are a lot of patients who advocate and who want it, hopefully that will spread this knowledge and interest beyond just a few academic centers that are really invested in the technology and understanding it. And now with a lot of the data that the companies have access to, like NeuroPace and Medtronic, it would be a fantastic industry, academia, research collaborative to understand what the signals are doing on a much larger, like hundreds of patients, hundreds of thousands of patient scale.
I like those hands. I mean, it's a hard question what you asked. I think it's sort of maybe partially what this conference is about. I mean, there's this question of how much do we need to do educationally, how much do we need to do versus advocacy, or is the tech just not good enough? You know what I mean? And so it goes back to the engineers and the people who are pioneering translation. And I maybe the second part of it is true as well. I mean, DBS has been around for a long time, but maybe it's been begging for adaptive technology, and that's what it's needed. And same thing, with prosthesis, that the fact that we need, to integrate these other harder technologies, these sensory things. So sort of blame the technology, not just the education. The both are clearly important.
All right. Another question that I had on my mind, it's probably, again, relevant probably to all of you, but I certainly saw it very clearly in Dr. Bixen's talk. Like, you need to think probably a lot about factors that go beyond just research, right? Thinking about like bringing this technology to like actual people or to the clinic or to everyday life. So, and again, that's probably a huge question, but like, could you say just a few words about like what are the actual like the biggest challenges that you're facing, like bringing this technology, these approaches from the laboratory to the actual like people to use it, or clinicians to use it, or people at home?
I mean, I think one thing, and I think this is true for all of us, is we need to recognize, you have the sort of the academic front, and then there's industry. And I don't think anything that we, ultimately when you get to patients, ultimately labs don't build this technology. We like to think, okay, we're academics, we do everything, but we don't build medical devices. We're not qualified, regulatory-wise qualified to bring medical devices. So that's one thing. I think it's you have your industry here. I know these other speakers must work very closely with the companies that they deploy. Maybe that's something to think in mind about how to be more open to those academic industry collaborations and figure out how to do that ultimately to the best benefit for patients.
Yeah, I completely agree with that. On the device side, that's not going to be my role of trying to improve the device and making a device that gets implanted perfectly every time and gives you the best signal quality. But I think what we've seen, kind of as I've already said, is the reliability requirements are high. And so a lot of what we're trying to do in the lab is try to push the science, try to understand behavior, see how that's impacted by sensory feedback. But the first products need to be something that works every single time in every single situation, no matter if the person is sleepy or on a different medication or in a different environment. And so I don't think that first generation of products is going to have the degree of function that you see from different academic research labs, but it's going to be based on those scientific findings and collaborating with industry to deliver something that is really robust and performs well.
Yeah, and the scalability is a huge question, and I think like Medtronic is running into that now with the deployment of adaptive. Like, how do we actually use it in patients? And even a center like as accomplished and well experienced as UCSF, even our group is having challenges in like who's the best candidate? How do we track this? And it involves a lot of patient engagement. It's not like we implant the device, we turn it on. It's like a light switch. It takes we're distilling like months and years of data intense collection with like postdoc students analyzing it to figure out what works for that patient. But if you're trying to do it on a mass scale, that's really challenging. So, and then Medtronic, they have to now hire, PhD neuroscientists to kind of figure out that problem as well. So yeah, I think it goes both sides.
Okay, perfect. Thank you so much for all your answers, for all the wonderful insights. I think with that we're gonna.
Sorry, can I?
Yeah. Do you have another question? Yeah, please. Go ahead.
Okay. Hi. I'm Melanie from the University of Oldenburg. My question is mainly directed towards Dr. Wang, but maybe Dr. Collinger and Brixen, you also have something to add. So I was very intrigued by the swimming study because I also did EEG during swimming, so that was something nice to see. So of course I'm also interested in swimming, and so I was wondering whether it might be a therapy approach as well to like take out the gravity from walking and to like either walking in the water environment or also just swimming to try to improve like motor function in patients with movement disorders, because I think there are also studies reporting that some Parkinson patients can like cycle when they're like on a spinning bike. If we take those motor function that are still somehow still present in those patients and try to train them, that maybe functions like walking might also be improved over time?
Yeah, that's definitely a really interesting idea. I think I think there are physical therapy rehabilitation programs using like aquatherapy for patients not only with Parkinson's but with like injury and neurological diseases in terms of motor function recovery. And I think one of the things, actually I trained a physical therapist in my lab who completed a PhD. It's just, we do all these therapies and there's no neurological basis. So the effects are often short-lived and it's not targeted. So I think one of the main motivation is like if we can understand what specifically within the brain circuit is disabled and whether we can train that circuit to prevent freezing, preventing falls, that would be a lot more fruitful, I think, to couple like rehabilitation with an understanding of targeted neuroscience approach.
Thank you again. All right, I think with that we're gonna finish that session. Thanks all our speakers again very much.